come to these LAM conferences over the years and participate with this vibrant and wonderful community. And certainly this event this evening will only enhance my appreciation for these events. I've been working for some time because I'm on the LAM Scientific Advisory Board. I actually saw the poll where my name come up for voting, and so I had advanced knowledge that this might happen. But and so I was working on a routine to talk about why me? Why did they choose me? But I've decided I'm not going to go into that. Except to say, I finally figured out why they chose me. And it's because they realize that I'm still young. And I've got a lot more to do in LAM research. So I'd like to give some thanks. First of all, I'd like to thank my father and my mother. My father, from whom I inherited his cleverness and his interest in humor. My mother, from whom I inherited her work ethic and interest in studies. I'd also like to thank my older brothers who were fond of sort of torturing me when I was about 10 years old by making me do long division problems to enhance my mathematical abilities. I'd also like to thank my wife and my children who've been inspiring. But then I'd also like to thank a list of people that I've had the pleasure of working with over the years, and to state that it's very clear that whatever I accomplished, I did not do alone. And so people that have trained with me and been very important members of my lab over the last 15 to 20 years are the following. Some are present, some are not, but I'm going to state their names anyhow. Lisa Hensky, Rocky Anda, Yolanta Kwiatkowska, Sandy Deborah, Hongbing Zhang, Penny Strank-Roberts, Nazreen Al-Hashemite, Geraldine Finlay, Piotr Kozlowski, Lindsay Meikle, Kristen Polizei, Hoshan Lee, Wei Chen, June Goto, Isabella Malinowska, and Ivan Chekoluk. And then finally, as I think you know, genetics has been my area of special interest and expertise, but it was a wonderful thing when we figured out that the TSE1 and TSE2 genes participated in this pathway. And I'd like to thank three individuals in the audience here who have contributed largely to my meager knowledge of signaling, those being Brendan Manning, John, and Lou Cantley here, who I continue to benefit from their collegial interaction. And then finally, I'd like to thank the funding agencies which have supported our work over the years. Tuberous Sclerosis Alliance, which has supported our research for now over 20 years. The NIH, of course, John Adler's Foundation, the LAM Foundation, and the LAM Treatment Alliance. So again, thank you very much for this work.
End of slideshow. That's never a good way to start. Okay. The title of my talk tonight is How do we define success? Cure, remission, or chronic suppression? The agenda is, I'd like to give a brief update from the Scientific Advisory Board. brief update of my biased view of LAM science advances, then a little bit about how do we define success. And the target audience is non-scientists, or 2,383 words and 2 glasses of wine into an awards presentation. That was before Sylvia added another 1,000 words or so that aren't on my copy. And 20 minutes from a dessert social. So in terms of news from LAM clinics, we have 2 new LAM clinics, one in Rochester and one in Boston. Lisa Hensky and Suhail El-Shamali are now the LAM clinic in Boston because Geraldine Finley has moved on to another career path. We have clinics that are now applying, which I believe will soon be approved in Kansas City, at USC, and in Montreal. And we have intentions to get multiple international LAM clinic sites over the course of the next year. New things in LAM clinics. We've got a LAM clinic listserv, thanks to Kim Hasselfeld, to share management advice. We have a LAM clinic registry that we're trying to get up and going. It's approved in Cincinnati and in Penn and a few other places, but it's getting there across the network. There's a LAM clinic tissue bank to collect serum and other samples for research. And the trail trial is currently being conducted through the network of LAM clinics. And if there's anyone who's potentially interested in this trial of letrozole for postmenopausal women with LAM, I'd appreciate it if you seek me out during this meeting. There's a new effort afoot to share the LAM clinic network with other rare lung disease communities. These diseases, Birt-Hogg-Dubé, Langerhans cell histiocytosis, Sjogren's lung disease, and lymphangiomatosis, are the most common diseases that are confused with LAM, and all of us clinic directors are becoming more and more familiar with these diseases, so we thought that perhaps we should just offer these disease communities the option of using this clinic network as well. That's been embraced very welcome fashion by all of these institutions, and in fact, if you look on the Birt-Hogg-Dubé website, they show our LAM clinic network and claim it for their own, which is exactly what we'd like to happen. And I mean that sincerely. We would like to see these patients. They're interesting to the 26 clinic directors that are scientifically curious people, and we're looking forward to focusing referrals in these disease populations to our clinics as well, and eventually letting them or helping them to share in these research tools that we're developing for LAM, including the registry, the tissue bank, etc. Now, there are several representatives from these organizations at this meeting looking into this possibility. So a few words about, just in the way of background, about LAM. I wanted to just review very briefly the steps in the development of LAM as a prelude to what I'm going to talk about. So the thoughts are that a LAM cell develops when a short circuit jams the mTOR growth pathway in the on position. LAM cells reproduce and form a tumor somewhere in the body, and we're not exactly sure where LAM cells come from. They spread from the tumor through lymphatic channels and target and accumulate in the lung. LAM cells make substances that dissolve and distort the lung. This is an image from Deacon Low that shows cystic changes in this coronal view of the lung, and here they're colored to make it a little more apparent. And you can see that there's a significant architectural distortion there, and that is something I wanted to make a point about in a moment. So there are 5 stunning truths about LAM I'd like to mention. That some of the best minds in science work on this pathway, and many are in this room. And because of that, progress in LAM has been absolutely thrilling. Now we understand more about the molecular basis of LAM than virtually any other lung disease. And I'm sure there are people who are going to disagree with me about that, but I get to present my biased view because I'm up here. And because of that, we now have an effective treatment for patients with symptomatic LAM. And the future is bright because there are many other ideas for potential treatments based on solid molecular understanding. So a few thoughts about progress in humans in this past year. I was just looking through PubMed since the last LAM meeting, and these are the things I found. Several groups have published that transbronchial biopsy is useful for the diagnosis of LAM, and that most women with tuberous sclerosis eventually develop cysts in the lung. That's a new finding from Lisa Young. Median survival in patients registered with the LAM Foundation is 29 years, which this study was done by Brent Kinder. It's the first population-based cohort. It's different different than the number you've seen on the internet because those studies were done in patients who present to hospitals and doctors' offices when they're symptomatic, and the LAM Foundation registers patients at all stages. And so across the whole spectrum, the median survival is much longer than what's been published. Everolimus is now FDA approved for brain and kidney tumors and tuberous sclerosis. And only a fraction of the cells in LAM lung lesions have tuberous sclerosis mutations. And that was a publication by Dr. Kwiatkowski in the last few weeks. That's a very provocative finding, something that I need to talk to him about and understand more about, but I think it's potentially important in the pathogenesis of this disease. Some promising lab findings. Estrogen blockage prevents lung damage from LAM metastases in mice. Statins are promising as combination therapies with sirolimus in mice. And the switch that is stuck on in LAM not only controls protein synthesis, but also glucose usage, lipid synthesis, DNA and RNA synthesis. These are some of the landmark publications that have cited LAM Foundation support since the last LAMposium. I'm not much of a sports fan, but to use a baseball metaphor, every one of these is a walk-off grand slam in a scientific career. Dr. Manning's lab that published a paper in Science that was recently broadcast across the LAM network about the role of mTOR in pyrimidine synthesis. The Sabatini lab published a paper in Nature about signaling molecules called RAG. Lenis lab has a paper accepted now just in the last few days in Cell for glutamine addiction that LAM cells demonstrate. Sabatini lab published a paper in Cell about regulator. In Nature by the Sabatini lab on mRNA synthesis controlled by mTOR. And in Science Translational Medicine by the Krimskaya lab about statins in mice. So this is a fantastic history of accomplishment for the LAM Scientific Board that have chosen these grants from among the 25 or so that have been submitted. I just wanted to, I don't have time to read all the names, but just acknowledge the effort of these folks who donate dozens and dozens of hours to review grants and get it right so that we can not only contribute to the body of knowledge for LAM, but biology in general. There's also a clinical science board. Several members of this board are in the audience tonight. So the next steps for LAM include efforts by the Foundation to obtain FDA approval for sirolimus in LAM, to complete some open trials. There's a SAIL trial that's enrolling in Boston. Dr. Suhail Elshanali and Lisa Hensky are the PIs for this trial. It's a combination of hydroxychloroquine and sirolimus. They're here tonight. If you are having interest in learning more about that, please look them up. And the trail trial that I mentioned with letrozole in postmenopausal women. There are trials coming. Dr. Vera Krutskaya is in the process of developing a trial for statins plus sirolimus in LAM. So I wanted to talk a little bit about how we view success in LAM. And to start with, I just want to mention where we are with treatment, and this slide has been shown several times already this meeting, but I wanted, for those of you who were not in the scientific sessions, I just wanted to point out that what the MILES trial showed was that for patients who were treated with placebo for a year, in the MILES trial, the loss in lung function was about 10% over a 12-month period. For those who were treated with sirolimus, lung function remained stable. When the drug and the placebo was stopped in the second year, decline resumed in the sirolimus group and paralleled that in the placebo group. The number of patients in this year declined substantially, and the scientific power of this arm of the trial is less robust, but nonetheless, we do believe that, like in many other models, that this treatment is suppressive and not curative. It doesn't kill cells, it suppresses. And that held true in this trial as well. So when I polled the LAM clinic directors at our most recent meeting at the American Thoracic Society, we talked about who we're treating based on this trial. All of us are treating patients with sirolimus, depending on their wishes, with patients with symptomatic LAM. We're also treating patients who have rapidly progressive LAM, sometimes even before symptoms develop or lung function becomes abnormal. We're treating patients with problematic chylus problems, such as chylus effusions or ascites, with sirolimus as well. But we're not treating patients with early disease who are asymptomatic if they're not progressing rapidly. So on the average in this trial, MILES enrollees have advanced LAM, and the one question becomes, can we extrapolate the results to patients with milder disease? Lung function remained stable only while the drug continued, and the question becomes, would the benefit of this drug be sustained over long periods? And finally, serious side effects were balanced between the groups, but can this drug be taken safely over many years? And I have Melanie Putnam's permission to use her case as an example here. This is Melanie and her 2 boys. Melanie had one of the more rapid courses of decline in lung function after a pregnancy that I'm familiar with. She went from an FEV1 of about 3 to an FEV1 of about 1 over the course of about a year and a half. When she was placed on sirolimus, there was stabilization in her lung function, and I think many of the LAM clinic directors in the audience could draw curves like this for patients they've seen in their clinics. So how do we define this response? Is this a cure? Did we see a restoration of health? Is this a remission? Is this a partial or complete eradication of all LAM cells? Or is this chronic suppression? Or is this restoring normal or near normal behavior to LAM cells without eradicating them? Well, just to define cure, and I wish I could have found a less controversial example of this one. Everybody knows this one. Lance Armstrong had widely metastatic testicular cancer in 1996. Robin Roberts had breast cancer diagnosed in 2007 and underwent aggressive treatment and then developed myelodysplasia in 2012. And as an example for chronic suppression, Kareem Abdul-Jabbar has a diagnosis of chronic myeloid leukemia, and he's now 5 years into treatment with good success. So could we use the term cure for Melanie's course? Cure to me means a complete restoration to good health, including normal exercise tolerance. And that may be possible in early LAM before the lung is damaged, but it's not likely possible in this year, this decade, in more advanced LAM where extensive cystic changes have already occurred. And that's because LAM has both fixed and reversible components that are variably responsive to therapy. Again, this is Dr. Lowe's image showing the cystic changes in the lung. What you can't see between the cysts are that this lung parenchyma is infiltrated with LAM cells. The cystic changes we feel are not reversible with treatment, but the LAM cells that infiltrate the lung can likely be tamed or killed with treatment. And this is the component that likely responded for Melanie when she was placed on sirolimus. So this response for Melanie was not a Lance Armstrong cure for LAM, since her lung function, though stabilized, was still quite reduced. The definition of remission for LAM, this is a little backwards, so I'm just going to put them all up here, is eradication of LAM cells. This will likely require combination therapies, and his usual regimen is an induction with strong treatments followed by maintenance therapy with less toxic treatments. And strong treatments can have consequences. For Robin, she developed myelodysplasia as a result of her breast cancer treatment. The outcomes from inductive treatment for remission include partial remission, where some or all LAM cells might be eradicated, and complete remission when, partial remission where some but not all LAM cells are eradicated, and complete remission where all LAM cells are apparently eradicated. Since Robin's complete remission lasted for more than 5 years, her breast cancer can be considered to be a sustained remission or cured. And sustained remission based on eradication of all LAM cells may be the most appropriate definition of cure for LAM and the most desirable approach. Well, how would we even know if all LAM cells were eradicated? Unfortunately, we don't have imaging tools or validated biomarkers to estimate the total body burden of LAM cells or to know if they're still there. For other cancers that